So thanks everyone for having me here, and I really enjoy getting to visit, and it's the first time I've been here since the new building is built and people are going in. So I look forward to meeting with many people today, and I'm happy to have a nice full crowd. Are there more seats available for some of the folks? You guys? I guess not. Oh, okay. So though this is a seminar, this talk will be a little bit more like a colloquium in that it's going to be an overview of a lot of the applications that one can do, particularly in terms of magnetic sensing and imaging using nitrogen-vacancy color centers in diamond. There will be small amounts of physics as we go along, but a lot of kind of laundry list of applications. My main message I'd like to try to get across to you is, for those of you who haven't worked in that field, is what makes these NV centers so good for magnetic sensing and imaging, and what are the types of applications that are already coming to fruition and what may be possible in the future. So I'm going to give some context and background along those lines, then a survey quickly of some of the recent and emerging applications, then go through the basics of magnetic sensing and imaging with NV centers in diamond. That's one of the most important things, try to just get the basic ideas across rather than a lot of the details. Hi, Ellen. And then go into a little more detail on a few specific applications, just some selected ones, and then maybe some talk about coming attractions, what I think might be possible in the future. Okay, so starting around 2008 with these papers that were published here, there's this new field that opened up based upon a lot of excellent work that people have been doing for many years before that, studying color centers, particularly nitrogen vacancy color centers in diamond. So for those of you who aren't familiar with the field, and I'm going to try to emphasize the basics, we are not using gemstones when we talk about diamond. And these color centers, as I'll talk about more, are little atomic scale defects, which have properties which are atom-like in many ways. The key properties include that they have an electronic spin. The spin can have long coherence times, even in room temperature diamond or diamond that's at a wide range of temperatures. And they're optically addressable, and you can read out optically. So typically, one is going to be illuminating the color centers with a green laser. It doesn't have to be green, but that's just what's convenient. Nice 532 nanometer laser. And reading the red fluorescence that comes out over about 100 nanometer range from about 638 nanometers into the red of that. And the intensity of the red fluorescence is going to be an indicator of the local magnetic field strength locally. I'm going to tell you about how that's done, but that's the basic experimental methodology. Green light in, intensity of red light out, tells you what the local magnetic field is in proximity to either one of those centers, all sitting by itself in diamond, or if you want, you can have diamonds in which there are many of the centers. So you can do nanoscale measurements with atomic scale individual centers, or ensemble measurements with many of them in the diamond. And given the current technology and the way that diamonds can either be grown to have these centers or they can be implanted, you can choose in a stochastic way what your average density NV centers are and kind of where they are in the diamond. You can have the diamonds be giant macroscopic centimeter or millimeter scale bulk chips, or they can be small nanostructured elements. There is still challenges in terms of being able to place these color centers exactly where we want them, but we're down now to the kind of few nanometer scale in terms of being able to place NV centers.
Hey Ron, how'd you get two publications that have the same title in the same month, different journals?
Notice this guy Lucan is part of both. Okay.
Sorry.
A non-serious question deserves a serious answer. Okay. One was the idea, and notice that had Jake Taylor as the first author, so that was the idea paper. And actually, that paper came, so now I'll try to be a little more serious. The ideas for doing this, at least from us, came late 2006 into early 2007. Misha and me, Jake Taylor, Paolo Capolaro, several other people, and that was outlined here. But it took a long time to get this paper published until we had the experiments working, and the other group did too. That's sort of a sociological fact, even though I think this paper is more useful, but kind of lays out the concepts. So this is a little bit of bragging, but I think it's true. There are many emerging applications of this technology, and I'm going to try to show you this as we go around and go through the talk. There are lots of applications, many of which are already having significant impact in some of these fields. Many more, like in the quantum information field, it will take some time. So this one chart hopefully is very simple and describes for you and explains to you what makes people very excited about using these NV centers as magnetic sensors. This displays approximate regions of magnetic field sensitivity, where the sensitivity gets better as you go in this direction, versus spatial resolution of your sensing as you get more higher resolution as you go in this direction, and approximate regions of sensitivity of different leading magnetic sensing probes. And it shows you that diamond can have this capability of being pretty good sensitivity at high spatial resolution. The ways you realize high spatial resolution, I will get into a little bit later, but one can go from a kind of near macro scale and have sensitivities that aren't too different from, say, squids or atomic vapor cells, although the best sensors are definitely still atomic vapor based, to having very high spatial resolution. And things like imaging the position of individual protons, single electrons, single proteins, single neurons, those aren't neutrons, that's single neurons, probing interesting questions in meteorites and rocks, and many other things have already been done. Not all of those results have been published, like these two have not, but they're working their way through the system. The other ones have been published. There are also many exciting applications in the condensed matter material science field, which are beginning to emerge. And I think that's an area in which I'm not really going to talk about in this talk today, except maybe one slide at the end, and I'll mention this a few times as I go along. I think that my own personal opinion about where the certainty of high-impact physics applications is highest is in this condensed matter field. Probing, there's such a diversity of interesting problems to study in condensed matter for which this combination of sensitivity and spatial resolution will be useful. I think it's going to be the most successful application in terms of basic physics. And so I would encourage you, my colleague Amir Yacobi, if at some point you want to invite him to come down and talk about that, he's really pushing hard into that direction, and I would encourage you to invite him to speak about that. So now, again, some more lists of highlights. So this is some of the things that have been achieved in the last couple of years. MRI of a single electron spin in a solid with less than 1 nanometer resolution. MRI of single proton spins on a diamond surface with about 1 angstrom resolution. I'm going to talk a little bit about that later. Imaging the magnetic fields that come from, and so when I say MRI here, I want to be clear, I'm not always referring to true MRI, that is measuring the, imaging the pattern of the processing magnetic resonance that comes from protons or other sorts of spins. Sometimes I'm more generally using it to save space here to indicate just magnetic imaging more generally. But in certain cases, like this one, we are detecting the Larmor precession of a single proton. Okay, magnetic imaging of magnetic fields that come from living biological cells with single cell resolution and subcellular resolution, measuring the temperature within living biological cells, something done by Misha Lukin and Hong Kong Park's group. Looking at tumor cells, which have been labeled with magnetic nanoparticles, and being able to do in a wide field, high throughput way, a bioassay where you can distinguish the tumor cells from normal cells. Other recent highlights, being able to do NMR and MRI, now I really do mean NMR, in this case, of samples which have more than 1 nuclear species, not just protons, but also something that might also have fluorines and phosphorus. 3 different groups at Harvard, Stuttgart, and IBM. Almaden did similar sort of work published very recently. Magnetic imaging of a primitive meteorite. What do I mean by primitive meteorite? These are meteorites which are, have fallen to the Earth and are thought to be unaltered since the formation of the solar system in the first few million years. We worked together with Ben Weiss's group at MIT and have been able to do what we believe to be the first determination of what the magnetic field was during the formation of the early solar system. That was published in Science a few months ago. Probing the genetics of biomineralization, an extension of the living biological cell work. There are theoretical ideas from Paolo Capollaro's group on how to take the single proton MRI work and extend it, very challenging goal, to extend it to be able to do single complicated biomolecule structural determination. I highly recommend people reading this paper. There's nice work done by Vincent Jacques' group in France looking at the nature of domain walls and ultra-thin ferromagnets, one of the examples of the early, what I expect to be large number of applications in condensed matter physics. Nice work by Misha Lukin and Hong Kong Park's group in probing nanoscale electron transport in metals, probing spin wave excitations in a ferromagnetic microdisk, work that's done by Amir Yacobi's group with my group helping out. So the list goes on and on and on, and I'm just scratching the surface. So after this list of applications flowing out, I think I'm hopefully making the point that in some very short period of time, a whole bunch of different things are getting done with the technology. At Harvard, many of my colleagues and I and our groups use NV diamond in various ways. I've mentioned Misha Lukin, Amir Yacobi, Hong Kong Park in chemistry, Marco Longcar in electrical engineering. Jeff Lichtman is a brain scientist with whom we collaborate. I'll mention some of that briefly as we go along. Evelyn Hu in material science. And we have many other collaborators around the world with whom we work in some of these technologies. And I'm showing their faces because bits and pieces of their work is going to show up in my talk as I go along. And there are more people than listed here, but I'm featuring these particular people because they're the ones whose work is going to show up at various points within my talk. I mentioned Paolo Capolaro already, Ben Weiss in the meteorites and early Earth rocks, Hako Lee at MGH for tumor cells, Arash Kameli in terms of cell biology, and others. Okay. So, for people who already work in the field of NV diamond, I'm now going to take a few slides and tell them some very basic things that they already know. For most of you who don't, this is hopefully going to get the basic ideas across. Again, I'm just trying to emphasize the basics. So here's a little cartoon of the diamond lattice in which two neighboring carbon atoms have been replaced by a nitrogen atom and a missing carbon or a vacancy. That is the NV color center. It also, to give it an electronic spin s equal 1, it typically picks up an extra electron from the lattice that's energetically favored. There is in the neighborhood of the N and V and the neighboring carbons a electronic cloud, which is energetically sits between the valence and conduction bands, the large indirect band gap in diamond. So it's energetically very isolated from the electronic excitations in diamond. And so one can think of this as just a localized atom-like quantum defect that has the optical and spin properties that I outlined before and are listed here. Electronic spin s equal 1, long coherence times. The T2 time as opposed to T2 star can be hundreds of microseconds up to a few milliseconds, depending upon the conditions you're operating in. If you use dynamic decoupling pulse sequences to isolate the NV, mostly isolate it from magnetic noise inside the crystal, and in the environment, one can get to a little bit larger than 1 millisecond. This is again at room temperature. There's a large Zeeman shift of the energy levels, the ground state spin levels. That's the source of the magnetic field sensitivity, that the energy levels move around rapidly with applied magnetic fields. Of course, diamond itself is transparent, it's robust, has excellent thermal conductivity and mechanical properties. It has this large indirect band gap, gives it a stiff lattice, it has a relatively small spin-orbit interaction, and it, for biological and other kinds of applications, is quite inert and biocompatible. So you can put living tissue in contact with diamond or make nanodiamonds and have them go into living cells, and nothing bad happens chemically. They can be, in certain ways, irritants if they're too large, but in general, nothing in terms of chemical interactions occur. I mentioned before that there's this optical spin state preparation readout. Tell you a little bit more about that in a minute. And one then, it can do regular old kind of ESR type manipulations with coherent or resonant or near resonant microwave, either CW microwave drive or pulses. Pick your favorite spin echo train or dynamic decoupling pulse sequence to be able to manipulate the electronic spin. And the system operates under a very wide range of conditions. Here's a little image of individual NVs in a diamond. So this is a particular example of one where the NV density is relatively low. So one can scan around with a confocal microscope and see individual NVs. They're single photon emitters and can be thought of as nice individual quantum objects. Here's a macroscopic diamond chip in which the NV density is so large that it's put through a filter here, so it filters out the green. But if one illuminates with a green laser, the whole diamond will glow red, particularly around the rim. Because of the large refractive index of diamond, most of the light is guided out around the rim. So in some of the bulk diamond magnetometers that we make, we're getting down to kind of picotessla per root hertz sensitivity, we will actually construct the diamond chip and build the detectors around the rim. You know, go to where the light is and put the detectors where the light is. That sort of thing. I'm not going to talk more about those sort of bulk magnetometers, but there's commercialization efforts going on them right now. Okay, now a few slides giving you some cartoon picture, not including all the details on the energy level structure, to again try to get the basic idea across of how one can do sensitive magnetic field sensing and through this optical detection technique and preparation. So here's the ground s equal 1 ground state triplet with the ms equals 0 level, and then the ms equal plus or minus 1 levels, separated by about 2.87 gigahertz due to, you can think of it as due to the magnetic dipole-dipole interaction between the two electrons, which make up this s equal 1 quantum defect, the NV center, splits it by nearly 3 gigahertz. Of course, these energy levels are going to be able to be shifted It's a regular Zeeman shift with a Bohr-magneton scale magnetic moment, which gives a very rapid change of the energy levels with applied field. There is separated up by energy a little less than 532 nanometers to electronic excited levels. There's a vibronic continuum that's associated with each of these group energy levels. And so if you illuminate with green, regardless of which energy level you start in, you can absorb a green photon, and one can decay down through fluorescence. And that process is radiative, of course, and is spin preserving mostly. So you can think of it as start in MS equal 1, absorb green, radiate in red, fluoresce in red, and you end up in MS equal 0 or MS equal 1. Wherever you started, you end up in the same level. There's also a pathway through metastable singlet states, a pair of metastable singlet states, a largely non-radiative decay path, which is non-spin preserving. So, and is favored primarily for the MS equal +1 or -1 levels. So one can, if one is in MS equal +1 or -1, there is a non-trivial probability to go down through this non-spin preserving path down to MS equals 0. So that, and here are some of the branching ratios and the time scales. So if you start out in MS equals 0, there's approximately a 10 nanosecond time to fluoresce typically with something like 95% probability. If you start out in the + or -1 levels, there maybe is 50-60% probability on about the same time scale to fluoresce and return back to the level you started out in. But on a much longer time scale, 300 nanoseconds, there's a non-trivial probability to go down to MS equals 0. So that means that if you illuminate with green and wait several hundred nanoseconds, regardless of which spin state you started in, you will always end up into MS equals 0. And then you'll be undergoing cycling transitions.
Why are these percents sort of not, like I would say look at the 10 nanoseconds or 100 nanoseconds, I would assign the percents based on that, no? Like why does it get to go this way if after 10 nanoseconds it's already down?
It's a probability per event.
But if after 10 nanoseconds it's already decayed down, I mean,
I'd say once it's here, it's stuck, and this is the time scale to decay once you're here.
I thought it was the upper one.
Yeah. Okay. In fact, there is I'm smoothing over some details. There's actually a infrared. There's a couple singlets in here, and there's an infrared photon that's typically emitted there too. But
so 60-40 is at the top.
Yes. So let me, okay. So here's what you do in a typical protocol. Start with your green on. Regardless of what state you end up in, you go via a form of optical pumping, not what we typically mean like when you're doing optical pumping in rubidium or something like this. It's not based upon the circular polarization selection rules. It's just based upon these branching ratios. After about a microsecond or so, one ends up in MS equals 0. That's your preparation step. Then step 2, spin coherence control. You can use either CW or pulses, but let's think of it in terms of a pi/2 pulse, resonant pulse. Put you into a coherent superposition, let's say of MS equals 0 and +1. Let's say we have a static field on that lets us select one of those levels, okay? Then now you're in a coherent superposition, and the electronic spin would just evolve in that static field or any other field to be sensed. It would accumulate some phase, and after some time later that you can control, you apply another pi/2 pulse, which will project along the longitudinal axis. By that I mean the MS equals 0, 1 basis, and then you illuminate with green again, and you can read out what your time-averaged fluorescence is over a time window, typically gated. See this comes on here. Come on, computer. There we go. A time window gated to give you the largest contrast between whether you were in initially MS equals 0 or 1 after the evolution in the field to be detected. And you can have this is your contrast here. This is kind of a typical single NV fluorescence data averaged over several cycles. You can average longer, make your signal to noise better, but you're not going to make your contrast much better than that. Contrast is actually typically worse when you have many NVs because there's heterogeneity between the NVs, and you have something I haven't mentioned yet, 4 crystal axes, and often you're aligning your static field along one axis. But that's basically your measurable that can be proportional to the phase accumulated in the presence of the field to be detected. So this kind of simple cartoonish picture, of which there is a lot more physics behind it, I'm trying to give you the simple picture, took a long time to establish. There was a lot of debate in the community, and this is all sort of pre-applying the NVs to magnetic field sensing. Getting the simple picture where we have a nice protocol that we can apply for sensing. There are many other important things that are going on in these systems, which I'm not going to talk about very much, except I'm going to list some of them here. And you should keep in mind. There are hyperfine levels. There's nuclear spin associated with the N that makes up the NV. That means it's typically not just these ms equals 0 plus or minus 1. You have hyperfine levels. You can take advantage of that with multi-frequency drives to be able to, yeah?
So I just want to make sure I understand. Your spin polarization step and your detection step, those look the same based on your description. But in both cases, you're exciting with the green. And
Right. So the difference, if I back up, is you're right. They're both qualitatively the same. The key is at what time period do you decide that you've either done initialization or you're detecting? You wait long enough, you end up in ms equals 0 in either case, right? In this time window, one could tell whether one is in ms equals 0 or 1 based upon what the fluorescence rate is. Of course, you have to calibrate things for your particular instrumentation. It's not, right? So that makes sense. So there is a very similar process qualitatively, but different in detail. And if I wrote down some of the pulse sequences, there would be the length of the green pulse that you put on, the gating of your detector, and things like this that would determine that. But it's a very, so it's, hopefully you see the difference, and you see that it's actually kind of simple in a way, which is one of the things that makes the system, and by simple I mean technically simple. You don't need multiple colors. It's just some gating and timing of pulses. Okay. Hyperfine levels I mentioned. Another key thing is that there is a composite spin environment. In addition to many NVs, you could have widely spaced NVs, but if you're at high densities, you can have NVs which can get to within a few nanometers of each other and can have dipole-dipole interactions. There are other both paramagnetic impurities, Ns. You may have a lot of NVs in there, but typically the conversion efficiency from Ns, from nitrogens into NVs, is on the order of %, 10%, that sort of thing. So there's typically many Ns for every NV. That's a frontier area of research. We'd like, in most cases, as few Ns as possible because they're sitting there, they do not have the nice optical properties. They are sitting there creating magnetic field noise, essentially, in your environment. Maybe you can think of them as interesting systems to couple to with your NV. Maybe they can be, you can eventually make a kind of spin chain between two NVs and couple them with Ns, maybe. That hasn't been done yet. But from a kind of simple magnetic sensing point of view, the Ns are unwanted and create background noise. Similarly, naturally occurring diamond has with natural isotopic abundance, has 1.1% of the carbons are carbon-13, which have spin 1/2. The rest are carbon-12, which have no nuclear spin. When I said spin 1/2, I meant nuclear spin 1/2. And so those also create a kind of magnetic noise in the environment. They can be a resource. One for very close NVs, which are close to the carbon-13s, which are very close to the NV, you can get strong hyperfine interactions. And in fact, you can do C0-like algorithms, which can take some phase that the NV has measured, let's say, in the magnetic field, map that information over the carbon-13. It has a much longer lifetime, and use that as a kind of quantum memory. So they're, in some cases, a resource. But it is also nice if they weren't there, because they will process around and make a kind of resonant nuclear spin magnetic noise that you have to account for in your measurement protocols. So can diamond be made with greatly reduced carbon-13? The answer is yes. So many of the samples, not all that are used in some of the experiments, people have synthesized diamond in the lab to have isotopically purified carbon. So it's almost all carbon-12, let's say 0.1% or something carbon-13 remaining. Sometimes it's kind of interesting, in an area in which people are working on now that I'm not going to talk about more, is actually to have so-called supernatural diamond in which the carbon-13 concentration is elevated, 5%, 10%, close to 100%. Kind of interesting to investigate what the energy level structure is there when the carbon-13 interactions with the NVs are dominant, and that's an interesting area too. But for a simple view of thinking as magnetic field sensors, the nitrogens and the carbon-13s, bad. They cause NV decoherence. So therefore, one often does dynamic decoupling to enhance the coherence times. So your T2 stars in the presence of this kind of, that is your inhomogeneous dephasing time in the presence of this kind of background magnetic noise that is primarily coming from these unwanted additional spins in the environment might be an order of magnitude or more shorter than the T2 decoherence times that one can get. And those decoherence times I'm talking about are just single Hahn echo decoherence times, and they can be hundreds of microseconds to a few milliseconds. And then if you put dynamic decoupling pulse trains on, one can increase often the T2 time much longer than that. Okay, there are also, I mentioned this, 4 crystal axes. So the NVs can exist along 4 crystal axes. There is some work going on in terms of being able to have the NVs grown into the diamond selectively, so with preferred orientation only in 1 or maybe 2 of the axes. But more generally, they are equally occupying, somewhat randomly, the different axes, or largely randomly. And so that can be, in many cases, particularly for ensemble magnetometry, can be just create background fluorescence. You are picking maybe 1 of the NV axes or maybe 2 and trying to measure the magnetic field projection along those different axes. And so there's additional fluorescence that's coming without much information from the unwanted axes that happen to be present in your field of view. But they also give you the opportunity to do vector magnetometry. If you reorient your sample or your static field or your field of interest along the different axes, you can work out what the different components of the XYZ components of the field for your sample of interest were. There's crystal strain, which creates inhomogeneities between the different NVs. There can also be, everything I've talked about with the S=1 NV center is the NV minus, where it picked up this extra electron from the lattice. That's energetically favored, but it's also possible with great shocks to the system, like very high intensity, energetic enough laser beams to strip that electron off and go to NV0. NV0 has S=1/2 and does not have the nice properties that the NV minus does in terms of the fluorescence that you get out depending upon the spin state. So the spin state dependent fluorescence that you have in the NV minus, you don't have in the NV0, so it's generally not our friend. Determining what your geometry of your sample is, how you fabricate it, how you process the surface to either attach samples of interest, or if they're very shallow NVs, a few nanometers from the surface, how to be able to maintain the good coherence times, maximizing your optical collection efficiency, all these sorts of things are important. This kind of repeats what I said before, but now you've got some sample of interest. You have, let's say, your NV center brought up very close. It can be extreme proximity. You have some static field that you've applied to split the levels. You also then have some additional, perhaps time-dependent or spatial-dependent magnetic field, and you can go through the same sort of protocol and measure that magnetic field through the fluorescence rate that one gets. How does one do that? There are different protocols. One can sweep through with CW ESR and sweep through the different transitions. One can run a Ramsey echo sequence for measuring DC fields. One can run these spin echo or dynamic decoupling trains for AC fields. As one particular example, here is measured fluorescence from a single NV at about 100 Gauss, sweeping the ESR, and you're seeing the two m=0 to -1 and +1 transitions. And you see that the fluorescence, when you're off resonance with your ESR, you're just staying in the m=0 level because you optically pump there, absorbing green, fluorescing red, and so the fluorescence is at a peak, at a maximum. When you drive with strong enough ESR on resonance, you reduce the fluorescence some because there's pathways to go through here with reduced fluorescence. And those narrow lines and the shifting of them with magnetic field can tell you what your magnetic field value is. So a rough expression of what this uncertainty in the magnetic field, and let's say whatever units you like, Tesla per root hertz, would scale like this. The gyromagnetic ratio, 1 over the gyromagnetic ratio of the NV, which is about 3 megahertz per Gauss. It's a number I keep in my head. And that's just given by Mother Nature and is a very large number. It's very nice because it's an electronic spin. The spin state dependent signal contrast that one gets, that is in most cases on the order, for single NV, on the order of 10%, sometimes a little bit higher. For ensembles, because of these background unwanted NVs, it's typically on the order of percent to few percent. So that's not a fantastic contrast, but it is something. Working to improve that contrast is another, by alternative detection techniques, is a current area of research. And then the square root of the product of 3 numbers, the optical collection efficiency, which is somewhat a function of the sample geometry and the experimental setup. N=1 for a single NV, but if you have multiple NVs being sensed, the ones that have the desired orientation that you're using for sensing, then you get better signal noise as the square root of the number of NVs. And it scales also as the square root of the T2 star time. And this goes to T2, or this extended T2 if one uses spin echo or dynamic decoupling sequences. Okay, there are many modalities for these sensors. And here's some little pictures and cartoons to indicate it. One can have nanodiamonds. These are obviously little red dot cartoons stuck in from PowerPoint, but it gives you the basic idea. They're usually not that uniform in size. It's a lovely EM image. One can have a scanning atomic force microscope, where the diamond nanopillar with a single NV a few nanometers from the surface and scan over a sample of interest. It's one way one can get this kind of nanoscale resolution. One can have a single NV just a few nanometers from the surface of a bulk chip and put a sample of interest on top, like a proton-containing or fluorine-containing sample in which you are going to be sensitive to a volume that's approximately the cube of the distance of the NV below the surface, the magnetic fields that are coming from that. One can have a bulk diamond chip, as I've mentioned several times, and a particularly common geometry that I'll talk about with several examples coming up is to have a bulk diamond chip, several millimeters on the side, with the NVs 10 nanometers, 100 nanometers, maybe a micron thick layer. So through the bulk of the chip, no NVs, and just a surface layer, a two-dimensional dense surface layer. You choose the thickness of that layer based upon the two-dimensional imaging resolution that typically one would like to have, assuming that one can use super resolution techniques as well as just diffraction-limited imaging techniques. And then you take the fluorescence and map it onto a camera. So you have a 2D array right onto your CCD or CMOS camera in which pixel by pixel you're getting information about what the sample's magnetic field is at the different locations. Hopefully that makes sense. So it's a two-dimensional magnetic field imager, which we often call the Y-field imager. Okay, so now a little detail, or too much detail, or not enough detail on perhaps too many applications given the time. I'm looking at the clock back there, and I probably have too many applications to go through, so I'm going to be selective as I skip forward. There are two ways in which we've done nanoscale NMR and MRI of samples of interest, in which this is supposed to indicate the proton spins or the nuclear spins in a uniform sample applied to a surface of diamond, and this is the region in which most of the magnetic field signal is coming to a single NV. So we can have a single shallow NV, a few nanometers below the surface, or we can illuminate many NVs and do a wide field image, in which each one has a little region that it's sensitive to, but if the 2D density of the NVs is high enough, you basically can make a wide field image where you're sensitive to small numbers of nuclear spins. So conventional NMR and MRI, of course, have inductive detection, macroscopic samples. They involve thermal spin polarization, typically in a high magnetic field. Here, of course, we're doing optical detection. We can have very small detection volumes. Statistical spin polarization. We could, in principle, put our sample in a high magnetic field and get a thermal spin polarization if you wanted to. But when you're dealing with very small number of total spins, thousands to hundreds of thousands or something like that in a kind of 10 nanometer on a side cubed volume, and you work out what the square root of that is and what your effective fluctuating statistical polarization is, it's often going to be much larger than the polarization one could achieve of nuclear spins in any tractable, practical static field. So we take advantage of this fluctuating, on average, non-zero statistical polarization of the nuclear spins to create the magnetization signal or the magnetic field which the NVs are detecting. This is an example of one of the pulse sequences. And let me skip over this. I can go back to this later if people want, because I've got, again, more things to talk about. Let me skip to some of the results. We are able to take the single shallow NV, put samples such as this PFOS material, which both has fluorines and protons in it, and measure the resonance spectra that we get from on the order of 100 polarized nuclear spins in the little volume of interest that we can detect as a function of static field, and see both the fluorine and the proton signal. And that's very kind of, from a chemist's point of view, very boring NMR spectroscopy. The thing that's interesting about it, we get the correct gyromagnetic ratios that are well known. The thing that's interesting about it is the small number of spins that we're detecting, on the order of 100. One can then use the wide field modality, create a mask in which this region here with an SiO2 mask elevates the sample about 100 nanometers off the surface, whereas here it's bare diamond, poor sample that has, again, both the protons and fluorines in it, and then do spectra as a function, or you basically acquire these wide field camera images. You can look at what the signals are and the spectra are at the different pixels and make a map of where the material is in contact with the surface and is not, and it agrees with what your mask provided for you. So this is essentially again, a kind of boring but non-trivial image of a material spread across the diamond surface, in which now we're going well beyond the spatial resolution that one could get with conventional MRI. So now, taking the next step, this is a nice experiment that Alex Sushkov led when he was a postdoc working with Hung Kook Park and Misha Lukin, which I was involved with this a little bit too. Alex will be starting in the fall at BU as an assistant professor. What we did after doing those experiments that I quickly went over before with a single shallow NV or a shallow layer of NVs and do these kind of boring samples is we also did another boring sample, but now going not to approximately 100 nuclear spins detected in an image, but a single nuclear spin, in this case a single proton. We did this by taking advantage of the dangling bonds that are at the diamond surface, these surface electronic spins, and coupling them to the shallow NV, which might be 5 or so nanometers deep. You could calculate what the, as I did before, if you had a sample filled with protons above it, you'd again have this kind of 100 protons you'd be sensitive to, because you're about 5 nanometers away from where all the protons are. Instead, if you have an electronic spin right at the surface, and you work hard to have very few protons, just the occasional proton here and there, you can have strong dipole-dipole coupling between the electronic spin at the surface and the NV, using a double electron-electron spin resonance pulse sequence. You're going to read that information out as to this coupling through the NV optically, and then you can use that electronic spin at the surface and its hyperfine coupling to localize protons on the surface to be the detector. So again, proton to be detected and imaged, coupled to electronic spin, which is going to be on the surface, which is on the order of angstrom or 2 away from it, hence very strong coupling. And then electron-electron dipole-dipole interaction between the NV and the surface spin to be able to determine the position of the electron spin.
How do you know how many electrons were on the surface?
Didn't until we did the experiment, but Hung Kook Park's chemical magic in terms of, and that him with some of his group members, Natalie de Leon, and some others did some remarkable oxygen and mixture of oxygen and other material plasma treatment of the diamond to be able to make the electron spins go from what we had, if you look at publications before this, a year or so before that, several of the groups, including ours and mine and Miri Kobe's and others, were seeing many of these electron spins at the surface at a high density. All right, there were many of them there. You wanted just enough so you can end up identifying their position, and it was a lot of that special treatment that Natalie and friends did that achieved it. And you can read more about that in the supplementary material of this PRL paper. Oops, I haven't put the reference up yet. It's coming. Similarly, being able to get the proton spins just right at the right density. Otherwise, you're overwhelmed by things. So by varying the static field, its angle, and taking advantage of the fact that the Zeeman interaction, the dipole-dipole interactions, the hyperfine interactions have different spatial angular dependencies, we're able to work out the relative position, looking down now, between the on orthogonal to the surface, between the single NV and different ones of these dark electronic spins. And then pick that one that's closest, this dark electronic spin, and then skip ahead, able to determine the relative position of this dark electronic spin to more than one proton on the surface. And this shows you the precision, and then these outlines here show you the accuracy of those measurements given systematic uncertainties on things like the NV depth. So able to, at least in this perhaps artificial, created, not very interesting yet to chemists or biologists situation, able to, at room temperature, map the positions of individual protons on the surface to kind of angstrom scale. All right, switching now from the nano to the wide field magnetic imaging modality. I'll rattle off a few of the applications and show you some examples. So this is this alternative with the macroscopic diamond chip, high density 2D layer in the diamond, where we're going to image the signals onto the camera. All right, this is a particular example of the manifestation for one experiment, one where we were studying bacteria, in which there's the diamond chip, here's the biological sample on the surface. We come in with the green light below the angle for total internal reflection, so it passes back out, excites the NVs in this region, but doesn't have the green light go through and zap the biological and kill the poor little bacteria. We collect the fluorescence onto a camera. We can illuminate either with a red LED just for kind of bright field imaging of where the bacteria are, in addition to the magnetic field imaging, or the blue LED when we're looking at fluorescence coming from dyes that are applied to the bacteria to study the sorts of things that biologists do anyway. That's one setup. Here's a picture of a different version of the same kind of setup. It's with Helmholtz coils around it that was used for doing studying of the meteorites. I'm going to show you some examples of shortly. my point is that these setups are relatively simple and easy to put together. They don't have that many components, and they can be reconfigured very rapidly. The current results I'll show you have normal diffraction limited resolution, but you can use super resolution techniques as well. So we did experiments and have done more than one round of experiment studying these amazing creatures called magnetotactic bacteria, which are believed to have evolved about 2 billion years ago and do amazing things like synthesize very regular, about 50 nanometer diameter magnetic particles. They align the magnetization vectors. They're prokaryotes, okay? They have no cell nucleus. They align them very well, tether them to proteins to the side of the bacterium body, and then it creates essentially a compass needle that they use for navigating down in the Earth's magnetic field to find the low oxygen containing water. In the north and southern hemisphere, the same species grow the magnetization things in the opposite direction. The uniformity of these particles is, I am told, better than humans can synthesize. And they're essentially solving a navigation problem with no cell nucleus, and they solved this 2 billion years ago. Okay, so there's a lot of, the biologists are interested, we use this as a model system because they're well suited for our diamond technology, of course. But the biologists are interested in this, and I've gotten interested in this too, because it turns out that many of the genes that they use are also in you, even you, Luis, even the highly evolved organisms. Even you. Why? I don't think we've got little compass needles in our head that we're using to navigate around to find the tea and the coffee and whatever. But it's thought that these same genes are regulating the synthesis and use of iron and other metals in the bodies for other purposes. So in that community, they are doing experiments such as purposefully either mutating these bacteria or knocking out certain genes, and want to see physiologically what this leads to in terms of the alignment of the magnetic nanoparticles, the behavior of the organisms, et cetera. There are existing technologies and tools that they had for magnetically imaging live bacteria using squids were not suitable. Didn't have the spatial resolution, and you had to do things on, if you really wanted better spatial resolution, since it's cryogenic, you had to use dead bacteria. It wasn't, it was a very nice early work, but it wasn't suitable for what they wanted to do. So we very quickly were able to take this diamond technology and apply it and be able to image the magnetic moments that come from individual bacteria in large colonies of them and watch them behave. Here's an example of a test where we took one bacterium and we could work out with our NV magnetic mapping the X, Y, and Z components of the fields, then go and take the same bacterium, now dead, find out where its magnetic nanoparticle chain is with EM imaging. Given the known properties of the magnetic particles, you calculate what the X, Y, and Z fields should be, and you find very good agreement, which was comforting, and said that we were likely doing reasonably accurate measurements of the magnetic field. Similarly, one could take the magnetic images, and here for 4 different bacteria, and work back out from them, for the X, Y, and Z, where the magnetic nanoparticle chains, these magnetosomes, should be, sort of an inverse problem, and then compare to what you see in the EM images and see pretty good agreement. So this was the sort of things that convinced the biologists that what we were providing was a useful tool. So that since then, working with Arash Kameli's group here, this is a very unscientific endorsement slide. I'm going to give a few of these because these are non-physics fields, and I have to kind of believe these people from other fields. When you study the wild MTBs, the magnetotactic bacteria, unmutated, versus the ones where they purposely mutated certain genes, which are, in this case, supposed to control not the synthesis of the particles, but the alignment of them to make long chains, we do find big differences between the magnetic moments you typically see in different bacteria between the mutated and the wild. And as Arash says here, they're very excited about this technology. They and other cell biologists who are now wanting us to build instruments for them see this as already a tool which is far better than the other tools that they had for the work in that community. So here's an example of which the sort of technology has gone rapidly from just physicists playing with it to other people wanting it and willing to spend money to have us build instruments and work with them. I won't try to go through all these words, but here, let's just focus on this cartoon. This is the example of the application in which you take rare cells, such as circulating tumor cells or pathogens, which have particular biomarkers on the surface that the biologists already know, and they know how to how to take nanoparticles, in this case magnetic nanoparticles, and label them with ligands which can go and find those biomarkers on the surface, but won't attach to normal cells. So then you would have magnetic nanoparticle tagged cells, if they exist, in a wide field of view, and normal cells which would not give a magnetic signal. If there were no such cells, you'd get none of those guys would show up with the diamond magnetic imaging. If there are, you could see them. This is the sort of thing people care about because they would like to know if these bad things are there. And they would like to know them. They don't want to miss any. They also don't want false positives. So they want a high reliability of this sort of technology. So here's an example over wide fields of view, millimeter field of view. Many cells where we purposely spiked in, the bright green guys, were cells which we put in at a certain percentage that both have the magnetic nanoparticle labeling, or we verified using the diamond, and had dye, a green dye so they could show up optically. And we found very good correspondence between where we see the bright green and where we see the magnetic signatures from the diamond imager. We've done this over several cell lines and several different types of biomarkers and find very good agreement. I'm not going to go through the details, but basically we agree extremely well with what's the current gold standard of optical flow cytometry. And so this is a promising technique which is currently being commercialized. And here's, again, another one of these endorsement slides from our collaborator at Mass General Hospital, where he says that he believes this technology will become widely used. We can also, here, let me emphasize, this is an example of a bunch of cells in human blood. There was one cell placed in with magnetic nanoparticle labeled. Optical techniques, you can't see it because it's a thick layer of blood, but since the magnetic fields penetrate through the blood, you can see it with the diamond imager. More applications, more endorsements in different fields because the technology is rapidly disseminating. So here is an example of one of these meteorites. It's an amazing story of a giant chunk of an asteroid which fell to the Earth. Initially, I thought, when these things fall to the Earth, don't they all get burned up? And it turns out the outer crust, if they're large enough, you can work it out. It's one of the problems I want to give in one of my physics classes, like widely applied physics, which Elizabeth took long ago. The outer layer will get burned away, but inside, if people go out there, they can find it's still cold. So by looking at the isotopic ratios, the chemicals and the materials that are inside of these meteorites, they can find in many cases that they have not been exposed to high heat, high magnetic fields, water, or other sorts of volatiles which would chemically alter them. Otherwise, these materials and chemicals inside would not be in pristine condition. And then they can go and slice these meteorites up and look in regions and try to find little grains which made of chondrules, essentially these early meteorites which formed in the first few million years as the solar system was forming, came together as little lumps of these so-called chondrules, which are on the order of tens of microns to a few millimeters inside, which are made of basically alloys of metal, which froze out at high temperature, and they're typically magnetized by at that point in which they freeze out, because they have a high coercivity temperature, they are magnetized in the presence of the magnetic field that was there at that point in time, and they remain magnetized with that value unless they have been heated up and altered in some way. And so when you go through and do the magnetic imaging of these meteorites, if you find, all the evidence shows that it's primitive and all altered, and you find similar magnetization in the different grains, but kind of randomly oriented, then that is strong evidence that what they were all, these things became magnetized in about the same magnetic field at about the same time, and then later came together and were gravitationally brought together and formed this meteorite over a period of time. And why do people care about what the magnetic fields were in the early solar system? It is believed to be a key player in the process by which angular momentum was shed out. Almost all the mass in the solar system ends up in the sun, and almost all the angular momentum ends up in the planets and other stuff that circulates the sun. But if you look at these gas dust clouds that exist just before stars and solar systems form, their mass and angular momentum distributions are more uniform. And so there are different competing models as to how this angular momentum is shed out, and they depend upon the order of magnitude of the magnetic fields. What we found, this is comparing the squid with its poor spatial resolution to what the diamond technology gives, is we found a value of a magnetic field which was consistent with the mid-range of the models, not the very high field models which indicate a kind of turbulent effects driven by magnetic fields, nor the very low magnetic fields in which magnetic fields would not have played a role. So this is just essentially one data point. We had many data points in the paper, but in the big picture, this provides one data point in the sense of one example from one meteorite. And now what these people want to do is go forward and apply this technology to multiple meteorites that will give them different time points and different the meteorites they know by the isotopic ratios, the distance away from the sun that they formed. This particular Semirkona meteorite is thought to have formed kind of between the Earth and Mars. Okay, so in fact, they're building up with our help instruments in their laboratory now.
So, excuse me, for this one, you sliced it with tomography?
Yes, they sliced it. Well, the part that we did, this is an existing field in which people were already doing this sort of work. What we come in and do is add the higher spatial resolution that you can get with the diamond magnet, and that was kind of simply shown in that image that I showed before. They could do coarser grain, sort of 100, 200 micron spatial resolution magnetic field imaging with squids, given that they don't want to make these things cryogenic, they need a standoff distance. And they were then having to do, of course, they could do higher resolution optical and other imaging to look at the different grains. And they knew there were bits and pieces which were smaller than their magnetic field imaging resolution. And they were claiming that it doesn't really matter. Our kind of ensemble average that we get from the squid kind of gives the right answer. And with the diamond magnetic imager, what we can do is have much higher spatial resolution to look at the individual grains. And at least in that meteorite, it doesn't change the answer too much in the final answers to the average magnetic field that the meteorite formed in, but it basically confirms it by finding that the sum of the different sources that we see in here is not giving an answer that's grossly different in terms of the ensemble average, but in other cases it may turn out to not to be the case. For example, applying it to early Earth rocks, as we're working on now, so this is work in progress, each of these little grains here that don't show up are known as Jack Hill's zircons that are formed more than 3.9 billion years ago. And the size of them and the magnetic signatures are small enough that the squid doesn't have the spatial resolution to study them. And they want to ask the question, are there little magnetic signatures inside of these rocks? For example, around this one, all the signatures come around the perimeter of the grain, which probably has to do with stuff getting between the grain and the rock in which it's embedded. So these 3.9 billion year old rocks are not a chunk. They're tiny little flakes of things, very small, that have over the billions of years been incorporated in other rocks. And they want to look inside these grains and see if there are signatures from materials that they can analyze whose would likely retain a magnetization which would indicate what the magnetic field was at 3.9 or 3.8 or 3.7, this sort of thing. Okay, so no other tool has the ability to do these sorts of studies. Whether we will end up finding anything useful is to be determined here, but I'm just giving you an example of how this technology is passing out into other communities. Okay, come on. Okay, so lots of applications. I want to now, towards the end of the talk here, give you sort of looking forward my guesses and my opinions about some of the kind of high impact applications, mostly in this bio area that I've been talking about. The condensed matter one, I'll have one slide listing some opportunities. So one is to continue this sort of single proton scale MRI I talked about before and really go to not boring samples, but complicated things like proteins. Will this be possible? You obviously have what I call the third dimension problem. We're able, under certain conditions, to map the position of individual protons in very special conditions right on the surface. Once you start having things which are third dimension, maybe it's not going to be possible. But there are some interesting ideas that Paolo Capolaro has come up with, and I don't have time to go into them here, but I would suggest you invite her down and give a talk on this. Really some cool ideas that have to do with taking advantage of knowledge. In most of these proteins, you're not starting with no knowledge. The things they're very similar one to the next with slight differences in certain regions. So you can use that knowledge to basically write down a Hamiltonian that describes the local interactions between most of the molecules with some uncertain terms. And you can take advantage of your knowledge of that Hamiltonian to basically study spin diffusion processes within the molecule, as well as couplings between the NVs and the dark electronic spins at the surface and particular spins. And then if you can orient the molecule at different positions and move it around, there was some hope to be able to reconstruct and fill in the gaps in one's knowledge. So you have a lot of known about these proteins, and yet some particular terms in the Hamiltonian to describe the local regions in a certain part of the protein that form a pocket, which is where a key biological thing can occur, is what's unknown. Big question mark about whether that will be successful, in my mind. Nanoscale functional magnetic imaging for brain science. Why don't I skip to it and say that we're already seeing magnetic signals from neurons. This has happened in the last couple of months. We're starting with simple worms, a delightful marine slime worm, which has a giant axon, and that's the little critter. And we can now see, for the first time, optical detection of the magnetic field of a single firing neuron sitting on the diamond surface. We have to signal average a few times. We have to make the thing fire. We're not yet imaging, so there's still a long way to go, but the results are only 2 months old. What we want to do is real-time sensing, not making it fire, of a non-trivial network of neurons that are doing something interesting, not necessarily a slime worm, but something more challenging. So we have technical improvements to go, but we can do it. We can sense magnetic fields from firing neurons. So it's an exciting time. I think there's going to be several years before we'll be able, a couple of years maybe, to be imaging culture mammalian neurons in an interesting circuit. Can we ever do an activity map of simple animals, particularly flat animals, or circuits like retina, things like this? Can we ever solve the third dimension problem without poking in? We'd like to do this non-invasively. To be determined. But at least for simple systems, we can study them. Another area, intracellular quantum sensors and maybe actuators to affect things using nanodiamonds. This has got a lot of potential to do things like, well, measure cell membrane potentials, control optogenetic tools. There are optogenetic tools now being developed in which the red fluorescence that NVs give off can control the optogenetic tool to itself control some protein to do things like turn on or off signaling through some membrane in cells or affect the delivery of biochemical cargoes. But an example of things that have already been done is what Michel Luc and Hong Kong Park's group did by taking nanodiamonds, using the fact that the zero field splitting, that 2.87 gigahertz splitting in NVs shifts with temperature. So you can monitor that and have different pulse sequences other than looking at the Zeeman shift. You can look at the splitting between the ms equals 0 and the ms equal plus or minus 1 together. And you can measure the local temperature inside of a cell, and then the applied heat to a gold nanoparticle inside of the cell, and they could, a modest amount of heat would warm it slightly, and eventually, as they warmed it more than 4 Kelvin, they could monitor when that particular cell died and verify that it was dying with fluorescence. And so now, after that demonstration, which was published a couple years ago, they are now going forward and using this to understand the expression, the sexual expression in C. elegans worms. They put nanodiamonds at different compartments within this worm, and they can study the temperature in different parts of the worm and selectively warm the worm in different regions. It is known that if the worm, whether it be is a hermaphrodite or a female or that sort of thing, is dependent upon its temperature, and that people have studied that in the past by changing the temperature of the entire group of worms. What they haven't done is change the temperature of different parts of the body of the worm and understand exactly what's going on to deal with that. So that's an example of, Michel Luc is doing biology now of that sort. And so I think there is a lot of possibilities here, and I think this technology will be successful. One could take slices of brain tissue from humans or animals. It's known that there are magnetic plaques that exist in people with diseases like Parkinson's and Alzheimer's. People can already sense the magnetic signatures from these kind of plaques at the kind of macro scale as you get up to many microns. But what's going on at the scale, at the smaller length scales at earlier stages of disease? The magnetic fields that we created by these plaques are very similar to the sorts of things that we're already seeing in some of these other examples I've shown you. So I think there's a high probability that this will work technically. Whether learning about the expression of these magnetic signatures earlier in stage of disease will actually help in developing treatments or diagnosis, other than just knowing earlier that you've got something we can't do anything about, is to be determined. But at least it's a new window in early stage of disease, and so we're planning to work on that soon. One can also take bulk diamond magnetometers, fiberize them, and make a network of MEG sensors on the outside of the scalp. This is already done with squids for doing magnetoencephalography, except squids, again, are cryogenic, and so they have to have larger standoff distance, which means they can't solve the inverse problem of where the sources of magnetic fields are coming from, as well as if you had room temperature samples on the surface of the brain. One could imagine doing this also in high field conventional MRI and have real-time structural MRI, functional MRI, and MEG sensors all together. So this is something which sounds good and might work, but there's some serious technical concerns with really making it work well enough. And this is one of these examples of something where we may get funded because it sounds so good, and the medical people want to do it, but I actually have concerns. I don't say this so loud to them, but I have concerns whether this will really work. Some of these things I'm enthusiastic about, some less so. The condensed matter area, I have a colorful slide here filled with lots of cartoons and some data. This, again, I'm going to repeat what I said before. This is a flourishing area, and I think there's a lot of physics to be done in this area. And I would say you should invite Amir Yacobi, and he can come tell you about those great things. Things I don't have time to discuss, or I'm not ready to discuss. Yeah. We have successfully now doing things which are analogous to conventional MRI, to do high spatial resolution imaging and sensing and manipulation of individual NVs across a wide field of view using strong pulse magnetic field gradients that we apply with microcoils. So this is a kind of Fourier or K-space imaging. Rather than rastering around and looking at individuals NVs in different places or wide field onto a camera, you can essentially acquire the data wide field, but do it in K-space and then take a Fourier transform if you like. That's working quite well. Improving the spin state contrast by taking advantage of the fact that with the right kind of illumination, one can take either the MS equals 0 or MS equal 1 states and map them into either NV0 or NV minus, which have very different spectroscopic properties, and use that to improve the contrast. There's lots of work in improving the T2, and as shown here, we're working on geometric phase as well, which we have data where we can get a much larger magnetic field range, and we're not stuck in operating with a phase evolution in one plane. Now we can drive and encode the information not just in the dynamic phase, but in the geometric phase. That works well to improve the B field range and to improve the DC magnetometry sensitivity. We are thinking about applications in a variety of areas of quantum simulation and whatnot, but that's one of the things I'm not ready to talk about, even though I put it on the slide. A lot of work in terms of being able to use Hartmann-Hahn techniques to couple the NVs to other spins in the neighborhood as a, hopefully maybe to do things like polarize the spin bath as a resource for some interesting spin bath physics and sensing. And there's also a sensitivity that we're also applying the NV magnetic field sensitivity through localized magnetic particles in appropriately elastic materials, which can be changed, whose distance to the NVs can be affected by pressure as a way to kind of have a transducer to turn the NVs into a pressure sensor and pressure imaging. So, and to conclude, on the coming attractions, probing early Earth rocks and meteorites, already working. I think it's going to continue to work. That's what the check means. It's already transitioning into this other field. Nanoscale probing of quantum materials, condensed matter physics, already working. This is highly likely to be a very successful field. Will we be able to do this single molecule structural biology? Don't know. Single neuron sensitivity, we've got it for worms, but can we really do useful things for brain science? To be determined. Intracellular quantum sensors, actuators, MRI, probably, although I gave it a question mark. Clinical tools, question mark. Scalable quantum information platform, big question mark. And so I'm at the end. I will show you a nice picture taken last fall before we were inundated with the snow, which many of you probably heard about in Cambridge with all the smiley faces and the list of names. Not everyone here works on NV-diamonds. Someone work on some of our astrophysics or other kind of topics in the group, and we get funding from lots of great places. I do want to mention, because I've listed them on the slides before, there's a small company in which I have a financial interest, and they have provided us with some funding, which has to be managed in the right sort of way, and is doing some of the transitioning of some of these bioimaging tools. So I want to acknowledge that I do have this financial interest, as I'm supposed to do, in all these cases. And there's some of the people in the company, and say thank you very much for your attention.